Thank you. I am very pleased to introduce tonight's speaker, Dr. Johanna Beam. We welcome her back. We welcome all our speakers, but especially she's a local kid made good. So, as a high school student in Longmont, she got interested in a meadowlark that didn't sound or look quite right. And she has a pretty amazing story about how this led her to study meadowlark genetics as an undergraduate, working with Professor Scott Taylor at CU Boulder and to contribute to all of our bird lists, or at least the possibility of our bird lists. So I won't spoil the punchline. I think you'll learn more about this story tonight. She went on to complete her doctoral degree in biology at Penn State, studying bird evolution. And she returned to the Front Range last fall as a postdoc with the Bird Genoscape project at Colorado State. And this is a very cool project that is harnessing the power of modern DNA sequencing technologies combined with field work. You have to catch the bird to get a feather to use for the DNA study. And what you get out of it is a map of the genetic variation of birds across their breeding range. So you can tie that together with their migratory routes and their wintering grounds and know what places might be especially important to protect from development or pollution or loss of habitat to preserve that diversity of birds. So I think you'll also learn a bit about this kind of work and or at least the general kinds of genetic work that go into this. As Bev mentioned, here at Boulder County Audubon, we go even further back with Johanna. She attended Audubon's teen bird camp at Hog Island, Maine, and took part in our teen naturalist program. And I have evidence of this with a lovely photograph that I found on our our beautiful Google Drive. She's holding I think it's a gold chick here. And to me, I love the way this photo connects to another piece of our mission at Boulder Audubon, Boulder County Audubon, because that you may not be as connected to. It's about providing early experiences that can help to shape somebody's path. So maybe one of the teens who takes part in our teen naturalist programs develops interest, curiosity, observing skills that lead them to become a lifelong birder or to become a volunteer with a local environmental organization. Maybe a person who is interested in nature has an experience at bird camp that helps them to find their tribe and to visualize themselves as a scientist or an artist or a conservationist. So as a chapter, we support these programs for young naturalists with our money and our time, not because we expect any particular outcomes. Not everyone becomes an ornithologist, but because of what can happen, the wonderful things that can happen when we bring people together for enriching experiences in nature. And so I want you all to remember that when the time comes to pay your Boulder County Audubon dues, round up to support those programs. Tonight, Johanna will share more about her journey as a birder, scientist, and scientific illustrator. I learned from poking around her Instagram account. She's a runner, a hiker, a photographer, a coffee drinker, a baker, a dog mama, and she likes to look at all kinds of natural phenomena, always with appropriate safety gear. So please welcome Johanna Beam. And Johanna, the floor is yours. Thank you. Thanks for coming tonight.
Thank you so much, Sandra. Can everyone hear me pretty well? Okay, cool. I may make a small adjustment. All right. Maybe that's better. Well, thank you so much for that introduction. It's so nice to be back here. I remember that the last time that I was here was probably one of the last in-person meetings that Boulder County Audubon had before COVID. So it's really nice to be back in person and see you all here for this as well. So I'm going to talk a little bit about art, science, and eBird and how it all works together and how I've used all of these different pieces in my work. So I'm going to start out by talking about almost none of those things by just giving you a quick overview of what I'm going to be talking about today. So the first is sort of my journey from being a little youngster birder artist to college student. Then I will pivot a little bit to talking about the meadowlarks of North America, as you've heard a little bit hinted at that I'll be talking about that. I'm going to talk about yellow-breasted chats. So this was the work I did in my dissertation, actually only a small part of what I did in my dissertation, but then also talk about how scientists like myself use a lot of eBird data in our work. And then I'm going to talk a tiny little bit about my career trajectory forwards and what I'm hoping to do next. So I'm going to start out from this very blank map that you see on the screen. I was born there. I grew up there. It might not look like much because I didn't put any state lines on the map, but that's eastern Nebraska. I did not grow up in Colorado, contrary to popular opinion, but I did eventually get out here. But as a kid, growing up in this area, there wasn't a lot of interesting things to look at. If you've ever been to eastern Nebraska, you would know that. I can say that because I grew up there. There are more interesting places when you go out further west, in my opinion. But I spent a lot of time reading and about all of these faraway, interesting places. And not only just reading, but playing outside. And that is me as a very young child. If you have been to Rocky Mountain National Park, you might know which lake that's at. We would come out to Colorado almost every year as kids. And I also would pressure my parents to get out of their comfort zone and take me places. So, having no experience in fishing whatsoever, I dragged my dad and my two sisters out to go fishing so I could catch a trout. It was very small, but very delicious. And then when I wasn't outside, I could often be found with some sort of sketchbook in the hand. And this is again me, I don't know, I was like 3 drawing what looks like a potato. That eventually evolved to birds later on. And I started out humbly, as everyone does, by drawing a northern cardinal. Maybe when I was like 4 or 5, I found that a few years back and I had to include it because it's kids' art. It's just lovely. It's fun. And then a few years later, I eventually moved to Colorado with my family. Don't worry. We all moved out here together in 2012. And being in the mountains and being in this area, I started to draw not only the things that were in my imagination, but the things that I was seeing. And so I started out with some colored pencil drawings. This was a blue jay I did in 2013. And some of my other backyard birds in Longmont. This was from a photo I took of a black-capped chickadee, and then I decided to do it in color pencil, and this was from 2014. And at this point in my life, I was homeschooled. I was sort of socially isolated, maybe a little socially inept as well in there, but I didn't know a lot of people who were interested in the same things that I did. And that's when I figured out that there was this wonderful thing called Bird Camp and a wonderful community that would help bring you there. And so I finally found out what these kinds of people were like when I was able to go to Hog Island, Maine in 2015, thanks to all of your generosity and scholarship in getting me out there. And this was the first time that I started to see that there was a realm of science that was focused on birds. And so this is actually Scott Weidenstall's hand right there holding a dark-eyed junco. And this was my first time that I ever got to see bird banding up close, and I was totally enthralled by it. And it would take me almost 10 years to be able to actually learn how to band birds myself later on. Any bird banding nerds, you can take a stab at aging this bird, and I'll tell you at the end what it is. And also, that photo comes back. I didn't know that original photo was going to be shown, but there's me holding a gold chick when we were out on field trips at Hog Island. And this was the time when I got to be around people who were interested in the same things that I was interested in. The people that I met, I still talk to this day. One of my fellow camp members, he is also doing his PhD, also in birds. And that was 10 years ago, and we still talk almost every single day. And I still see him at scientific conferences. So the connections that I was able to make at that bird camp have lasted literal decades, and will continue as I go forward, both in the connection to birds and the connection to science. And being a part of Teen Naturalists also fueled my desire for knowledge. As a weird homeschool kid, I would just dive deep into everything that I started to learn about. And through Teen Naturalists, I was able to go on interesting field trips, meet with interesting people, and see some really cool things. And so I got to see the sandhill crane migration a few times in Llewellyn, Nebraska, as part of a Teen Naturalist trip. And then bringing things back to the sort of Hog Island theme, this was a black-throated green warbler that I did in colored pencil shortly after coming back from Hog Island, and just feeling very inspired and excited about being a young birder and learning more about the world around me after being around all of these really cool young people who were doing the same thing. And shortly after I went to camp, I found my research sparkbird. So you probably all know about sparkbirds as being, the bird that sort of gets you into birding. My original sparkbird was a dark-eyed junco, but my research sparkbird that has already been hinted about was a Chihuahuan meadowlark. Now, it was not called a Chihuahuan meadowlark back then. It was called a Lillian's eastern meadowlark. And finding this bird at McIntosh Reservoir and seeing that it was a little bit different and getting really excited about it actually got me grounded from my parents. I went out to look at this bird too much, and then I went birding too much, and they grounded me. But that did not hold me back because then working with Ted Floyd, we put together this presentation, which I put in quotes as my first research presentation talk because Ted actually did most of it. I just stood up there and talked about meadowlarks for 10 minutes. But I was able to give this talk in 2016 as a high school senior on this apparent nesting record of a Lillian's eastern meadowlark, which was a subspecies of eastern meadowlark at the time. Now, this desire to study birds, that was sparked sort of by this original bird that I found in 2015, kind of led me to study biology and study birds at St. Olaf College. So I attended St. Olaf College studying or doing a double major in studio art and biology. And during my time there, I helped bring to life the St. Olaf Birding Club. I think there were like 3 members. It was a little sad, but we did our best. We had plushies. We tried really hard. And then during my time there, I got another exposure to bird banding, where I got to band my first bird, which was a northern saw-whet owl. The owl was not in college. I was in college, just so you know if you're reading that. Just a clarification. But to be honest, that meadowlark was still at the back of my brain, and I could not get it out. And I just knew that I had to do more. And when I was at St. Olaf, it was great. I loved studying art, but the biology part was just missing to me because all I wanted to do was to figure out what was going on with that Lillian's eastern meadowlark. So I packed up my things essentially and decided to leave St. Olaf and go join CU Boulder as a junior in college and study meadowlarks under Dr. Scott Taylor. And thankfully, he took me in. I interviewed with him, having never taken a genetics class. I had just finished my freshman year of college. I was thinking about transferring. I walked into his office and I somehow managed to weasel my way into his interest by saying that I wanted to study meadowlark genetics, again, with absolutely zero knowledge about genetics. I had no idea what I was talking about. But somehow it stuck. He took me on as a student, and then I was able to do some meadowlark research. This is a photo of me recording some Lillian's meadowlarks, or Chihuahuan meadowlarks, as they're now known. Yes, with a broken finger. I had dedication back then. So now I'm going to switch gears a little bit and talk a little bit more on the science side of things. So how eBird scientists and illustrators all get along. I don't work for eBird. I'm not here with big eBird. I'm just a scientist and illustrator who's really excited about being able to use all of this cool data that you all, and hopefully myself sometimes, are collecting when I get the time to go birding. So I am an evolutionary biologist. That is what my degree is in. And I have several big life questions, things that I am dedicating my life to try to answer. The first of which is sort of the mother of all questions, which is how in the world did the Earth get all of this crazy, beautiful biodiversity that we see today? So this is a tree of life. So this is a phylogeny for the science nerds out there that shows the evolution of birds across the globe through evolutionary time. And you can see that there's birds of all different shapes and sizes on this evolutionary tree. And that we can see sort of how birds have evolved from the beginning of time. So the secondary question to asking this, how did we get all of this cool biodiversity, is how in the world has it been maintained over time? So modern birds have been evolving for about the last 66 million years. And somehow, all along this time, we now have around 10,000 different bird species around the globe. How in the world is that maintained? What are the little evolutionary forces that are working to keep one species separate from another, or that drive certain species together to hybridize? And then we can break these questions down into some smaller questions that I've started to ask during my dissertation work, which is, why are some species beautiful and colorful to the human eye, like these tanagers of Western Colombia, and other birds not as much? So these are the warbling vireos, and I know there are a lot of people out there who love the warbling vireo complexes, and I do too, but they are a little bit less colorful than other species. So warbling vireos were recently split. So we have Eastern and Western, and then we have a variety of some other variation within warbling vireos below. So we can ask why they're colorful or why they're not so colorful. We can also ask, why do birds live where they live? How do they sort of decide where is a good habitat and what is bad habitat? How are these changes, or how are they affected by these changes over time through land use change by humans? And how does this impact their evolutionary trajectory, or how they interact with each other as maybe separate species or species that are coming into contact? And the truth is, the answers are not always very straightforward. And we kind of know this if you know anything about the yellow-rumped warbler complex, where they hybridize like crazy, where they meet in British Columbia and Alberta. But where else they are, they don't. And the answer is still not there. But in general, let's not get too far ahead of ourselves here. We have other species to talk about. So the first of which is going to bring us right back to my undergrad. So this was the theme of my undergraduate honors thesis that I did at the University of Colorado Boulder, or here essentially, because we're in Boulder now. And this was not the title of my thesis, but it is getting at what my thesis was, which is, how many meadowlarks are there really? And you probably know the answer to this, or you think you know the answer to this, but we will check that out. So first, I'm going to step back from the meadowlark question a little bit here and talk about cryptic species. Now, you might be wondering what a cryptic species is, and I'm going to tell you it's not that. That is a cryptid species. So it's not that one, but what actually is it? So a cryptic species is something like the warbling vireo complex, where these species may look almost identical, at least to humans, the human eye, but in their DNA, they aren't identical. And that means that when they are in the same habitat, they may behave as completely separate species, and they don't interbreed with each other or hybridize. Okay, that's great, but why does that matter? Cryptic species matter and are cool, because they're birds. I mean, come on. But they're also cool because birds have a different visual spectrum than humans. So that means that birds can see into the ultraviolet spectrum while humans can't. And so we might think that they look drab or boring, or they look exactly the same and are impossible to tell apart. But to birds, they might look completely different. We just don't know yet. The other reason why is that there was a recent study that came out last year that found that drab or less colorful or range-restricted birds are essentially completely understudied. People tend to favor birds that are colorful, that are bright, that are located near where their universities are located. And it means that a lot of these birds just have no science behind. We don't know anything about them. And if you have a Birds of the World subscription, you can go on there and you can look and it'll be like, yeah, we don't know. We don't know what it eats. We don't know where it goes. We don't know where it migrates. There's just a lot of data missing for some of these birds. And it's really sad because they're really cool, like the warbling vireos here, which I don't work on, but are very cool. And essentially, cryptic species provide insight into the quote unquote true biodiversity that the world has. So if we don't fully understand how many species there are in the world, how can we protect those habitats that those birds live in? How can we conserve those areas if we don't know, first of all, what's there? And so this is why cryptic species are really important and why I love meadowlarks, because they're also a cryptic species complex. So you've seen these before. They're the meadowlarks of Chihuahuan and Eastern and Western. But let's get into a little more interesting stuff here. So the Sternella meadowlarks are the meadowlarks of North America. There are other meadowlark species that are located in South America, but we're going to focus on these ones, which are also located in South America, but you get my point. These are the ones we're looking at. So the Western meadowlark, which is Sternella neglecta, inhabits areas from southern Canada all the way down into parts of Mexico. They're located here. They're here year-round, and you can hear them singing. They are one of my all-time favorite birds. We have Chihuahuan meadowlarks now, which I'll go into a little bit more later, that are located in the southwestern part of the United States and then go into parts of Mexico, all the way down to parts of Oaxaca. And then eastern meadowlarks. I'm only focusing on this part of their range, and I'll tell you why in a couple slides. But the eastern meadowlark samples that I have, I'll be focusing on, are from this part of North America. We don't actually have any samples from Mexico, so really we just have samples from the US. But their range actually goes all the way through Central America and into South America, and they also inhabit Cuba, which is very cool, and we'll go into that in a second. But first, I'm going to show you some genetic data. This is some plots. Don't get scared. I'm going to walk you through it. So we have our 3 species here. This is called a principal components analysis plot. It is a fancy term for just a clustering analysis. So you're going to think of this plot like a map. So if you have a whole bunch of points that are in one area, it means all those individuals are closely related to each other. And if you have a whole bunch of points in another area across the plot, it means that those are 2 separate things. So if you are with yourself, your points are together, you're with your like things. If you're far, you're different. Does that make sense? Kind of. So with meadowlarks, what we find is that there are 3 distinct groups. So we have Chihuahuan meadowlarks, which include a subspecies that we sampled called Aro pectoralis, which is found in southern Mexico. Western meadowlarks are completely separated on the other side of the plot there. See if I have maybe a pointer, maybe I don't, but are in brown on the top side of that plot. And then eastern meadowlarks are in their own separate group at the bottom right of the plot. Now, the important thing to remember here is that prior to this, we thought that this Chihuahuan meadowlark group was just a subspecies of eastern meadowlark. We thought it's just the same as the rest of them. It just happens to live in a very different area than the rest of these subspecies. But this group of eastern meadowlarks that I'm showing you here also contains several subspecies of eastern meadowlark that are found throughout the United States. But we don't see those other subspecies like the ones from Florida in a separate group, but we do see the ones that are in the Southwest US and Mexico as being a separate group. And we can see that they're all equally different from one another, which means this Chihuahua meadowlark, it's definitely not an eastern meadowlark. It is its own separate species. And that, if you've been paying attention to taxonomy, went into effect in 2022, I think. And Chihuahuan meadowlark was not my first pick for the name, by the way. There were better names out there. Because it doesn't only live in the Chihuahuan Desert, right? If you know things about the deserts of Mexico. It's a little sad, but it's okay. They did their best. They picked what they did. So coming back to this question of meadowlarks in North America, we now know what's happening in this part of North America, right? But we're still missing a good chunk of Mexico. So we know that there are 3 distinct species if you're from sort of the mid part of Mexico and on northward, but there are still a handful of subspecies of eastern meadowlark. I think there's about 8 of them or so that are named subspecies that we know basically nothing about. And those are of this group here. And what's happening with these populations? Is there a chance that all of these are their own species too? And the truth is, we don't actually know. So in my undergraduate work, we used about every data point, every genetic sample that was available south of the border of the United States. And there just were no samples. No one had collected a blood sample, a feather sample, or a tissue sample from some of those areas. And so we just have no idea what's happening. But we can use eBird data to look and see how different these meadowlarks may look. So we know that Chihuahuan meadowlark is a little bit different. Western meadowlarks are a little bit different than Eastern meadowlark. So maybe these meadowlarks look a little bit different as well. And the way that we can do this is thanks to both local eBirders like yourselves, who take photos of Western meadowlarks here, local eBirders in places like Mexico and Cuba and Colombia, who take their photos, and also birders like you who go on vacation to these places and take photos while you're there and then upload them to eBird, because that means people like me can go look at those photos and see what those birds look like. And a lot of scientific illustration prior to modern times was done using museum specimens. So they would go out and they would shoot the birds and kill them, and then they would study them afterwards. But in lieu of museum specimens, eBird photos make really great references for art. And so this is just a time lapse of a Chihuahuan meadowlark that I drew based on a selection of photos that I picked from people's eBird checklists that were taken and made within the range of Chihuahuan meadowlark. And the point here is to say that when you take a photo and you upload it, you are making available a ton more data than you think, because people like me who are interested in the differences of these various birds can look at what the birds look like where you found them, and then compare them to what birds look like from other places. And so we're going to take a couple examples here. So the meadowlarks of Cuba, for instance. This is an illustration I did of the meadowlarks, some of the meadowlarks that are in Cuba. And you might be thinking, okay, that just looks like a meadowlark. And you're right. They do all look really similar. But if you take enough time, you'll see that there are a few differences that I'll point out. Sorry for the people on Zoom, I'm pointing. But if you look at the yellow part of the breast and where you have that black V that comes down, you'll see that there's a whole bunch of little black streaks that form there. They're pretty small, but those don't seem to be present on most other Eastern meadowlarks. But for some reason, the meadowlarks of Cuba have those. So those might be a difference that is part of some genetic variation that we don't know about yet. And the same can be said for some meadowlarks in Colombia, like this one. So there are several different subspecies of meadowlarks within Colombia. This is just one. So this is just some of the local variation that you could see. But this meadowlark here, and so some of the meadowlarks down there, have really dark lores, so that area between their bill and their eye. And they also tend to have a lot of yellow in their malar, like a Western meadowlark does. But we think of them as Eastern meadowlarks. So it poses some questions about what could be happening in these areas where we don't have samples. But now we know that there could be something to look at because of photos that we can find on eBird. So ideally in the future, we'll be able to actually obtain some samples from these populations, ideally working with people on the ground to understand how different they are. There is some talk of some ongoing work with the Bird Genoscape project, maybe of getting to work on this in the future, but we'll see. It's a very cool complex, and we know that there's a lot more to be uncovered. So now I'm going to take you into some of my PhD research. So in 2021, I started my PhD at Penn State University, PhD in biology, and I was looking to study evolution in birds. Didn't really know what it was going to be. I'll give you a hint. It has to do with yellow-breasted chats. Now, you might be wondering what the story is with yellow-breasted chats, and I'm going to take you a little bit back in time. So pre-2017, the yellow-breasted chat was considered to be, and I quote, a weird wood warbler in the family Parulidae. So the family Parulidae is our wood warblers. It's things that you think of during migration, like Townsend's warbler, McGillivray's warbler, yellow warbler, common yellowthroat, and the yellow-breasted chat was thought to be in that group. And if you've seen a yellow-breasted chat, you probably know that they are much different than a warbler. They act very differently. They sing the weirdest, craziest song. And fun fact, they sing at night. During fieldwork, I have been woken up by meadowlarks at 2 or 3 a.m. singing. I don't know why they're singing then, because they should be asleep like the rest of us. But they do some crazy things. But then in 2017, these yellow-breasted chats were split from the wood warblers and put into their own family named Icteriidae, which is the worst naming scheme ever because we already have a bird family called Icteridae, which is the blackbirds. So the truth be told, we don't actually know what's going on with yellow-breasted chats, whether or not they're a warbler, whether or not they're a blackbird, or whether or not they're something completely different. But we have some other questions that we can answer. So I'm going to take you to this range map first. So this is the range map of the yellow-breasted chat. The sort of main orangey color here is the breeding range. Migration is in yellow, and the non-breeding range is there in blue. There's also a small resident population in purple. The yellow-breasted chat has two subspecies that have been named and studied. The first one is Icteria virens auricolis, which is known to be, it translates into the yellow-green golden collar, if you take its Latin name verbatim. But it's found in the western half of North America, from Canada all the way into parts of Mexico. And then we have the eastern yellow-breasted chat, which is Icteria virens virens, which just means yellow, green, green. And that is because they are greener overall. And they reside in essentially just the United States, from Pennsylvania sort of eastward to Iowa, or westward to Iowa, and then down south to the Gulf. So there are a few morphological differences in yellow-breasted chats. They are also a cryptic species. You might notice a theme here. But let's walk through some of these differences in their plumage. So the first of which is they have this thicker white malar, or this white sort of moustachial stripe there, while the eastern birds have a much thinner white malar. They also are generally more grayer on the head and back in the west than they are in the east, where they are more green and a little bit more brown on the head and the back. And then finally, they have a thinner bill than the eastern birds that have a thicker bill. And I did debate David Sibley on this. I am right. They do have the western birds do have a thinner bill. He did not believe me on that, but I had the data to back it up. So continuing on with our story here. There are a few different ways we can look at the range of these chats. So first is, again, this range map that I've already shown you, but I'm going to bring in some eBird data here to show you a little bit more nuance into what's actually going on with yellow-breasted chats. So this is eBird observational data. So these are just sightings. This is from the year round. So there are birds in the wintering range there in Central America and birds that breed as well. But we can see that those two range maps don't really coincide with each other. There are areas where people have found yellow-breasted chats that don't appear to be within the bounds of the range map on the left. But then, if we break that down even further and we look at the eBird status and trends, so this takes our eBird observational data, runs some models on it, and then finds where the highest density of yellow-breasted chats are, or any species of interest, and models where those are in geographic space. And we actually see a very different pattern where it seems like these yellow-breasted chats have a very contiguous habitat out east. So they have one big blob where they're found, but then once you move westward, it's not so contiguous anymore. You have these riparian valleys that come up. You have river systems. They actually have some habitat changes in what they prefer in the west. Sometimes they prefer scrubby habitat, like in the foothills around here. You go up to Montana, you only find them in riparian systems. And then you go down to New Mexico and you find them in a slightly different kind of habitat. And so you can see that it's very patchy. And there's also this interesting pattern occurring between the east and the west. So I'm going to zoom in here. This is just the breeding range of yellow-breasted chat now. So I've taken away migration and the non-breeding area. And we have this gap between the east and the west. And we don't know what's happening in this gap because we know that there are two subspecies, but are they meeting each other here? Like, what could be happening? And so we're going to actually take to eBird first. And we're going to look at all media types for yellow-breasted chats on eBird during the breeding season. So the question here being, can we tell the subspecies apart using eBird photos. So we've already used eBird to sort of identify potentially interesting populations of meadowlarks, and now we're going to do the same for chats. So we're going to first off, and this is actually what I have done, I'm taking you step by step through my research process. We're going to take this individual in western Kansas. There is a photo available for this bird, and we are going to dive in. So what subspecies is this particular individual yellow-breasted chat? I'm going to give you some hints. It has a large white malar, and it has a gray head and a back. So what subspecies does that make this one? A western, yes. So now we know, okay, we might be able to identify these birds based on eBird observations and photos. So now we can ask this big question of whether or not these subspecies overlap, meet, or touch at all in the Great Plains, or are they completely separate with different habitat preferences? And so this was a small project that my partner and I took on in the span of I think it was a single afternoon. And we went through all of the eBird photos that are available and tagged them on whether or not we thought that they were a Western chat or an Eastern chat. And so what we have here are birds that we thought were potentially Western yellow-breasted chats are in blue, and birds that we thought were Eastern yellow-breasted chats are there in red. Points that are purple were sort of ambiguous to us. It kind of looked like they could be either. At that point, we had no idea what was going on, so we just labeled them as purple. And then gray were points that we weren't able to use to identify because the photos were maybe a little bit too blurry or a little bit too far away. But we still wanted to make sure that we captured and used every single photo that was available at that time for this region of the Great Plains. So if you take a look at this, you'll sort of see this dividing line. And so it seems that there is probably not a whole lot of overlap, at least based on our eye on what we think these subspecies are. There's not a whole lot of overlap when we use these eBird photos to try to determine range edges in birds. So now we can sort of take a step back and think, okay, if we think these might be separate things that don't meet with each other, are there potentially any genetic differences that have been cooked up between these two different subspecies over evolutionary time? And we can use some fairly non-invasive methods to get bird DNA now. So if you are sciencey at all, you might take interest to know that unlike human red blood cells, bird red blood cells have a nucleus, which means they have DNA in them. Our blood cells are not nucleated. And this means that we can take a blood sample from birds and get a really, really high quality sample of their DNA. And that means that we can catch and release these birds so that we're not actually doing any harm to their populations. And so this is me holding the first yellow-breasted chat that I ever caught. I was very excited about it. And this was in Texas. And so our goal here, my goal and my field assistant's goal, was to comprehensively sample the Great Plains, try to get at this question of whether or not these two species could, or two subspecies could possibly be coming in contact. And because I'm both a scientist and an artist, I wanted to quantify all of the possible ways these chats could be different from one another. So in addition to taking a blood sample for their DNA, I also took a lot of comprehensive measurements like bill depth, and this is how I know their bills are different from one another, is because I took all these measurements, things like their wing length, their tail length, their leg length, and then all of the different measurements of the bill. In addition to that, I also took feather samples to look at whether or not they were differently colored. And using these feather samples, we could tell whether or not they had different colors according to the bird's vision. So this was not a subjective thing like, oh, I think that's a little more orange there, but actually using modeling to show what the bird sees based on these exact spectra of the bird coloration. So here I'm going to bring back the range map. The point here is to show you that on a range map, where you're used to seeing the breeding range, the non-breeding range and stuff, and that is how things are differently colored on a range map. But here, I'm going to show you a genoscape. Now, you've heard the term genoscape. It is the genetic landscape or the landscape of genetic variation. And so what this shows us is how many different genetic populations they are within a species and where geographically they are located. And so this is the genoscape for yellow-breasted chats, which show two very distinct genetic populations which happen to align almost perfectly with our boundaries of the subspecies. So in blue, we have the western yellow-breasted chat, and in green, we have the eastern yellow-breasted chat. And I'm going to bring you back to the same plot that I showed you, excuse me, with the meadowlarks, where remember that if you're more similar to each other, you're going to group together, and if you are different, you're going to group far away from each other. And this is a good way for us to look at all the individuals and how all the individuals are sort of separated on space, because we saw how they separate on a map, but maybe there are some other things happening here at a finer scale that we can look at. And so what we see here is, again, two very distinct groups. And we also find this other pattern. So this, again, supports this hypothesis that there are two separate, at least, populations happening. But I'm going to take this away and show you something else. So again, in the west, these points are blue. In the east, these points are green. And then we have points in the middle that are pink. These are what we call potential hybrids. So these fall in this genetic space as completely intermediate between our two genetic populations. So they represent individuals that have come as a cross between two different parental types, in this case, a western yellow-breasted chat and an eastern yellow-breasted chat. So you might be wondering, okay, I've heard hybrids, I've seen maybe some hybrids, it's probably not a big deal, but let's talk through some hybridization stuff real quick. So some species, including some species we see around here, hybridize a lot. This is the case for bullocks and Baltimore orioles. This is taken from a paper that was published in 2020. This is the contact zone here in that box between these two distinct species. And we find that despite them being two very distinct species in the west and the east, respectively, in the middle area, which spans from here along the Front Range all the way into parts of Nebraska where I grew up, are these hybrids. And they not only form a lot of hybrids, but they form a lot of different phenotypic variants of hybrids, which basically just means there's a lot of variation there in what they look like and whether or not they look like their parent that was a Bullock's oriole or a parent that was a Baltimore oriole. And that essentially means that they've been hybridizing for a really long time. And this hybrid zone is pretty stable. So it's not changing a lot. These hybrids are viable. They're able to interbreed, but they're not taking over the population. They still act as distinct species overall. So we have species that can hybridize a lot when they come into contact. We can also have some species that rarely hybridize. And this is the case with these black-headed grosbeaks and rose-breasted grosbeaks. This comes from a paper that was published last year. So in the yellow, we have the range of the black-headed grosbeak, and in the red, the range of the red-breasted grosbeak. Rose-breasted grosbeak. And then in this little inset, we have a map of South Dakota, which was the sampling transect where these two birds meet. So this transect that they sampled ran from the western side all the way to the eastern side, from one species range all the way to the other. And the hybrids were only found along a transect of about 61 kilometers, which I think is somewhere around 50 miles. And that is a much, much smaller area than the hybrid zone that we saw in the previous slide, which ran from here to essentially Omaha, which is about 580 miles. So some species rarely hybridize, like these ones, where they sampled 138 individuals and only 9 appeared to be hybrids. So what does it really mean? What are the possible outcomes of hybridization? And hybridization has several different outcomes. The one that most people know of is the mule. So if you cross a horse and a donkey, you get a mule, but that mule cannot interbreed. It is an evolutionary dead end. It stops there. Nothing more can happen. They have two distinct species that interbreed, and their young are sterile. We've also seen a fairly stable hybridization, as seen with these yellow-rumped warblers, where they do hybridize quite a bit, but the hybrid zone doesn't really do anything. It's a similar story with the orioles. It's a fairly stable hybrid zone. It's going to exist for a long time, and it has existed for a long time. And then one other possible outcome to hybridization is that you can have hybrid speciation. This is the case with this helianthus sunflower, where you have two parental species that hybridize to form young, and eventually, over time, those hybrids become totally isolated from their parents and no longer interbreed with their parents whatsoever. Now, hybrid speciation essentially never happens in birds. It's only maybe been characterized in a few cases, but what we mostly see are evolutionary dead ends or stable hybrid zones over time. And what we find in yellow-breasted chats is that there are very, very few hybrids, and the hybrid zone is minuscule. So I'm going to bring back up this genoscape, and then I'm going to highlight this area here. So this is in the middle of this. This is Kansas. You have Nebraska to the north, Colorado out to the west. And what we see is this is our hybrid zone. This single point is our hybrid zone in central Kansas. And this corresponds to Sand Hills State Park. It's a 2 square mile state park. It is very small. I have tromped all the way through it. And in this area, we only found 5 birds in the entire park. And I went there 2 years in a row to try to look for these birds. And the important thing is that as soon as you go a little bit to the west of Sand Hills State Park, you have birds that are entirely genetically Western. They show absolutely no ancestry of Eastern. It's like Eastern are thousands of miles away. And then you go a little bit to the east of this hybrid zone, so a little bit to the east of Sand Hills State Park, and all of the birds are entirely Eastern. They have no Western ancestry whatsoever. But somehow, in this very small little state park in the middle of nowhere, Kansas, there are hybrid yellow-breasted chats. And so this is to show that hybridization in yellow-breasted chats is really, really rare. It only happens in one area. It is not widespread whatsoever. So it has a very similar evolutionary story to what's happening in the grosbeaks, where they tend to act like entirely different species with no interbreeding really happening between them. And so in summary, yellow-breasted chats should be split into 2 species, which is... I was grilled on my committee whether or not I supported this because apparently I didn't say it clear enough in my dissertation. So I'm telling you all now, yellow-breasted chats, it's coming. There's gonna be 2 species out of them. So you might be getting an armchair lifer if you've been to the east and seen the eastern yellow-breasted chat. And I don't know what they'll be named yet, so don't ask me. I don't control it. But if you have any really good ideas on chat names, I can probably put a pretty good word in once we get the proposal written. So that was sort of the summation of my PhD. Again, that was a small part of some of the work I did. But to bring it back to the timeline, I graduated with my PhD in December 2025. So that was just last month that I went to graduation. And now you can sort of think about what's happening next. So give you a hint. Shades of yellow, brown, black, and white. Has a worldwide range. I'm going to be working on the horned lark. So it's a very fun bird. They are found entirely around the globe. And I'll be working on things very similar to what I've worked on in my undergrad and during grad school, which is things like geographic variation. So the very first goal will be is to understand these worldwide genetic differences and morphological variation in horned lark. So this is just a small snippet, again, of the variation that is found within the entire species complex of horned larks. So some of them are very yellow, some of them are entirely white. Some of them have actually a lot more black to their bib that comes down. And they are found as far north as very far north in Canada and very far north in Russia, but also found in places in isolated ranges like in the middle of or just south of Bogota in Colombia, and also found in places like the Atlas Mountains in Morocco. So they are very cool species to work on. And the main part of my work will be to try to look at whether or not these populations of horned lark are specially adapted to their environments. Because even here in Colorado, we have horned lark that you can go find out in Pawnee that are breeding there, but we also have horned lark that breed on the alpine tundra. So I know that these birds somehow are able to survive in really, really dry, arid deserts, but then are also able to survive at 14,000 feet. So there are a lot of questions there about whether or not we can find these genomic signatures of local adaptation. And so this is a project that I'm doing as my official title is the National Science Foundation postdoctoral research fellow. It's a lot of words. The main thing is I'm a postdoc, so I get to do science full time. It is my dream job. And I work in collaboration with the Bird Genoscape project up at Colorado State in Fort Collins, and then also at Texas Tech with the Mantha Lab down there. And you can see these are the three species that I've worked on. Meadowlarks, horned larks, and yellow-breasted chat right there. And this is the artwork that's actually in some of the offices that we get to work in, which is awesome. So in summary, to bring it all home, meadowlarks currently comprise 3 distinct species across North America, but there is likely a lot more variation to be uncovered in Central and South America. So if you have any people down there who want to collect some meadowlark samples for me, let me know, so I can do more work on meadowlarks. And then photos and videos that you all submit to eBird can provide really critical evidence for us as scientists to understand the geographic variation that is present within these species and some species that I have worked on, like meadowlarks, chats, vireos, and horned lark. And then finally, yellow-breasted chats likely form 2 genetically distinct species where hybridization is rare. So watch out for your life lists because probably got a new one coming for you. And with that, I would like to thank all of the amazing people that have gotten me to this point, including the Taylor Lab where I did my undergraduate research, the Taves Lab where I did my PhD research, and then all of these amazing collaborators who have helped with samples and getting the analysis done and trying to figure out what is going on with that little hybrid zone in yellow-breasted chats. With that, I'll take any questions.
Wow, that was fun. So let's see, we'll start with questions in the room. Carol. Oh, yeah. Oh, she's got one.
Since I'm pretty much an auditory birder, I'm curious as to the chats, the Eastern and Western. Are they different sounding? And would that help you to, say, I mean, sort of like the meadowlarks clue you in? It's like, oh, this doesn't sound like a Western meadowlark. Yeah, because they are very different.
Yeah, so kind of, yeah.
Does that help at all?
Yeah, so in case you couldn't hear the question, it's do the yellow-breasted chat subspecies sound different from one another, and can you use that to identify them? To my ear, no. I can't really tell them apart. There was a master's thesis that was done in the 2000s that did show that they have different characteristics in their song, so that maybe a computer can tell them apart. To my ear, they sound the same. There are maybe some sequences of their song that are different from east to west. They seem to be able to tell each other apart because when I was in that hybrid zone and I was trying to call them into our nets, I had to play both Eastern song and Western song, and they only came into Western song. So they do seem to be able to tell each other apart somehow. But if you find a really good way of telling them apart, please let me know because I have not figured it out yet.
Other questions in the room, and then we'll go to Zoom. Go ahead. Oh, wait, let's get you a microphone.
So do the North American species of meadowlarks interbreed? Are there hybrids? And if so, where?
So not really. So our genetic data didn't show any evidence of hybridization throughout sort of evolutionary time, which means that probably for the last few millions of years, if they do interbreed, it's locally and they don't last for very long. If they do, they do breed in captivity. So that was the work of Bud Lanyon back in the 70s that found that they will breed if you put them together. But for the most part, hybrids don't really occur, maybe in very, in very rare instances. And if they do, it's usually in places like, I think the hybrids have been found in the 70s in like Wisconsin or Minnesota between eastern and western meadowlarks. But to my knowledge, there's not really been great evidence of like Chihuahuan and western meadowlark interbreeding or Chihuahuan and eastern interbreeding.
Do we have any questions on Zoom? Then we'll come back to the room.
Yes, we do. Karen Holweg.
I think you've just answered this, but let me go back and make sure I've got it right. In the hybrid zone, you find birds with DNA from both the western and the eastern bird combined in that hybrid, right?
That is correct, yes.
And that hybrid bird is not able to breed with an eastern bird and is not able to breed with a western bird.
So we think that they might be able to breed. So within my sort of 5 birds that I sampled, we think that 4 were sort of viable hybrids. So we think that 2 of them were first generation, which means each of their parents was a full parental Eastern and a full parental Western.
Yep.
So there were probably 2 of those. There were 2 that were 2 of those F1 or first generation hybrids that were together. And then there were some back crosses, which were hybrids mixed with a parental. So we think that they probably can, but we would need a lot more samples. And we don't know if the hybrid zone can support that many more chats than we already sampled there.
Okay, do hybrids breed with other hybrids?
Yes, they do. Yeah.
Okay.
Yeah. That is generally what we found, is that they will breed with other hybrids.
Thank you.
Yeah.
Sandra, is there another Zoom question?
Yes. Nellie, would you like to unmute and ask your question?
Yeah, sure. Thank you for this presentation. I was just wondering what name you would have opted for the Chihuahuan meadowlark.
So this might be controversial, but I really liked pallid meadowlark. That was my vote. When they asked me to be part of the English naming proposal, you had to like put what your top choice was, and mine was pallid. Because Chihuahuan meadowlarks are really pale overall. They are the palest of the meadowlarks, and that is probably because they live in a desert environment. But the committee said that there weren't enough birds named pallid to make it worthwhile. And if they were named pallid, they weren't on this continent. And that was their reasoning why. And I don't agree with it, but it wasn't my choice. So I thought something to go with the actual like phenotype, what the bird actually looked like, would be better than the place that only half of its population lives in the Chihuahuan Desert. Yeah.
Okay,
Yeah, I'm glad you agree.
We have a couple more questions in the room. Jim?
How long does it take for new species to occur? And in light of the book of The Beak of the Finch and the recent things on the radio about quick changes in beak size during COVID in Los Angeles and stuff.
Yeah.
And sort of overall, that book seemed to suggest that there was a balance between trying to be colorful to attract mates and not be colorful to be attractive to predators. Right. How does that all function? Is it the ecosystems are changing in such a way that sometimes they need brighter colors or duller colors based on predators? And maybe a final thing is with insects, they say there's a lot of these one-to-one dependencies on a pollinator and a plant.
Yep.
And are there similar things, like some birds are pollinators and stuff? Is there a way that things can evolve out of that? Like, what do you do if aren't any more of those rusty, whatever they are, bees around, and you still need to?
Yeah. So I think you could write a whole dissertation on all of those questions very easily. So I think the first one is how fast does a species form? Generally, we think on the order of hundreds of thousands of years for a divergent lineage to form and be what we call reproductively isolated, which just means it's not interbreeding with another population that it was previously interacting with. In birds, we generally think hundreds of thousands of years to millions of years for a new species to form and be stable over time. So there are times when there may have little changes like beak size. I think there was that paper on dark-eyed juncos in California where they had beak size changes during COVID. And that is more of the plasticity. So all of the variation that you already have in your genome that can change a little bit over time to adjust to the environment around that bird. And then I think your final question was like the evolutionary arms race between plants and birds. And that it is very hard for an animal to get out of that. And it would probably take, again, the order of thousands of years for something new to evolve, for an individual to adapt to a new environment. But that's why we study local adaptation now. So there is always the potential that that can happen faster if there's a lot of extra genetic variation that can be used when a bird faces new stress. So maybe faster, but as far as we know, it's pretty slow. Yeah.
Did I see another question back here someplace? Okay, one more here. We'll go back to Zoom and then back to the room.
Hi, I was just curious if you were familiar with the hybridization of the mountain chickadee and the black-capped chickadee up here.
Yeah.
And has that been studied a lot that you know of? Has it been continuing? Do they interbreed, or is it similar to what you expressed with the meadowlark?
Yeah, so the question about black-capped chickadee and mountain chickadee hybridization is really cool. It's been studied extensively by people in the Taylor lab. So I never studied it. I think they're still studying it now. It seems like the reason that mountain chickadees and black-capped chickadees hybridize with each other is actually due to humans. So human disturbance within Boulder. So this was work done by Dr. Catherine Grabenstein, who's, I think she's at Cornell now, but she was a grad student at the time when I was an undergrad. And she found that human change to the environment has allowed those two chickadee species to come in contact with each other and hybridize. And that also the use of bird feeders all the way up into the mountains has actually attracted black-capped chickadees into habitat where they never were before, and also leads them to hybridize. And so actually at my parents' house, thanks to bird feeders, I found hybrid chickadees up there. Yeah.
Sandra, do we have more Zoom questions?
We do. Jason, would you like to unmute and ask yours?
Sure, thank you. I just am curious, when we see the plot graphs that show the genetic differences, like the one that you showed of the chats, it seems reasonable that there would be genetic differences with like two isolated populations. And I'm curious if there's a rule of thumb for like how different quantitatively they have to be before you'd consider them separate species, as opposed to just subspecies or something else.
Yeah, so it's all human defined. So we are the ones who define species, so we got to kind of set the boundaries. We used to sort of play by rules of mitochondrial divergence when we used just mitochondrial DNA. But with whole genome DNA that we use now, there's not exactly a rule of thumb. But if your genomes are at least a few percentage points different from each other in a way, we sort of count them as potentially as different things. And it depends what other data there is that we can look at to sort of identify whether or not these are different populations. So if we thought that they were two populations that maybe had a what we call a cline of genetic variation. So it would be like in the orioles, where you see that their hybrids they kind of look from one species and they gradient to the other. We can see that with genetic data sometimes, and that just means that they're not all different from each other. They still interbreed all the way throughout. And that is basically all to say that there's not really a rule of thumb. We just use a bunch of other data in addition to that kind of PCA plot, that pointed plot that I showed you there to look at. So it's not just that. We look at the evolutionary, how long it's been over evolutionary time. We think that these two species have been separated from each other. You can model habitat over time as well to see whether or not they have actually been in contact, because the habitat we see now is not the same as it was 10,000 years ago or 500,000 years ago, et cetera. So maybe that answered it, but there's a lot of nuance. I don't want to go too much into the weeds.
Sandra, do you
Thanks.
have one more, and then we'll come back to the room?
Yeah. I have a question, which is on your maps showing that the chats only hybridize in that little state park. How do you, it's one thing to say, I didn't find any hybrids anywhere else besides that state park, but it's a really different thing to say there aren't any other hybrids anywhere else. Can you talk about how you know that you didn't miss some somewhere else in another small plot of land?
Yeah, so using eBird data, maybe I can go back to this. I don't know if I can go back fast enough. So basically using eBird data, I looked at all the potential areas that chats have been found. Just give me a second here. We'll get there. So going back to this is essentially where all the different spots that chats have been found, or we can go back even further to look at this one.
Michael, can you pop that up on the screen so we can see it too? Can you share it, or is it?
Yeah, so if we look at the eBird observation map here, if you were actually to zoom in on this, you see that there are not actually a lot of places where these chats occur or people find them. A big part of that is just that this area is underbirded. So not a lot of people go to the middle of nowhere in Kansas to try to look for birds. But that is also because there's not a lot of available habitat in central Kansas anymore, or in central Oklahoma, where we also sampled, and in parts of Texas, where we sampled. So we went out and we looked at basically every tract of land that I could get access to. So in order to ban birds in these areas, we do have to get local access from either landowners or the state or county government to go out there. So there were some places that I tried to sample and I never heard back from anyone. And so we couldn't go out there to look for chats. But I looked and I looked and I looked, and I spent many days scouring all of these spots, both online to look for available habitat and in the field, boots on the ground. And chats just aren't there. So they just flat out do not actually inhabit habitat in some of these areas. And so it seems like, yes, there could be other areas where these chats hybridize. Maybe there's another unsampled hybrid zone in Texas. We would need some more data to find that out. But it seems like for now, based on the fact that even if you go a little bit to the northeast of that hybrid zone, that none of those birds have any Western ancestry, that the hybrids are really, really, really localized. Because if you saw that they had some Eastern or they had some Western ancestry into the Eastern habitat, then we would know that the hybrid zone is a little bit bigger, but we don't see that really at all. So it seems like it's probably the case. We can say with fairly good certainty, maybe not 100%. But yeah,
Thank you.
hopefully that helps.
All right, I saw Ted's hand up. Carol's coming to you with the mic.
Back to Carol McCaslin's question about the vocalization. So there's a grad student, Isabel Bush, up at CSU. She's not in your department though, who's just developed these wild computational methods using like sort of souped up principal components analysis to separate bird vocalization. So she's shown that common and Chihuahuan raven vocalizations, although our ears can't make much out of it, they're really different from each other. They scatter widely. They're just broadcasting in parts of the frequency range that we don't hear. So but I would think that applying that to the incredibly complex of songs of chats would be cool. By the way, her thesis defense is on February 2nd up in Fort Collins. Anybody wants to go see that. But she's going to be talking about ways of quickly identifying vocal differences that our inept human ears can't really resolve. So it all has to be done computationally now. We think we can hear bird song, but we really don't. But computers do a really good job of hearing.
They do. Yeah. Oh yeah. I know. I think that's probably true with yellow-breasted chats. My guess would be that, if you really tore apart their song, you would find differences between the East and the West. I just didn't have the heart to do that during my PhD. I had too many other things going on. But it's an open project for someone to look at in the future for sure. And hopefully maybe that's a better way of identifying them because they do look very, very similar.
Do we have more questions in the room? Bring you the mic.
Hi, Joanna. Great presentation, by the way. I don't know if you remember, I asked you a question over email a few months back about my mystery meadowlark that I
Yeah.
have down in San Luis Valley. Actually, I have a question though about these chats.
Yeah.
Because I had no idea you were going to talk about chats, but you mentioned having found hybrids in only one location.
Yeah.
And it was a state park that I actually went to this year
Oh,
during my own field work
Yeah.
for the Conservancy last spring in May.
Mhm.
And yeah, and like what you said, yeah, that park is like the only natural looking spot within a long distance. That's why I
Yeah.
stayed there, because it
Yeah.
was the only place I could camp.
Yeah.
But my question has to do with those chats, because I just checked my checklist and I saw that I had 3 yellow-breasted chats.
Oh.
Excuse me, on my 2-hour walk.
Uh-huh.
And of course, at the time, I didn't think much of it, but I'm now wondering, well, gosh, maybe I had one of those hybrids.
Yeah.
But I don't know if you mentioned this in your talk, but I was curious of the hybrids that you had. I mean, how many non-hybrids, I guess, or were you finding there? Like, what are the chances that I may have actually had one, which of course I have no documentation for?
I would say almost 100% that you found a hybrid.
Okay,
So
interesting.
like I only sampled 5 birds in the 2 years that I went there to look for
Okay.
chats.
Okay.
One of them appeared to be, and they were all of hybrid ancestry. All 5 that we had. So we didn't find any pure Eastern or Western birds in that location at all.
Wow.
It was only hybrids, and they just were of different sort of stages in their hybrids. So some had already been crossed with other hybrids. One had been crossed with a parental. Don't know where that bird came from. It's hard to say, but yeah, I would say about 100%. So
Wow. Okay.
it's really cool that you've been there, because not that many people have been to
Right,
Sand
yeah.
Hills State Park in Kansas. But the other thing that I will mention, since you've been there and you've seen that it's like the only cool attractive habitat in a
Right.
while, is that we don't actually know if there are a ton of ecological differences between the 2 different, populations. So there is a chance that that maybe hybridization was a lot more common in the past, but only this attractive habitat exists now, because Kansas has gone through and has a really long history of agriculture and farming. And the vast majority of the state in the last 100 years has been turned into agricultural fields. So there are only a few areas where chats exist anymore, because they are like micro habitat specialists. They need a very, very specific kind of habitat in order to set up a territory. So some other fun facts there.
Sandra, more Zoomer questions?
We do. Kevin, your turn to unmute and ask your question.
Yes, so I saw a lot of art throughout the presentation and a lot of digital art towards the end. I was just asking if you have a favorite art medium, and does that change depending on what you're creating? So kind of research versus just for fun.
Yeah, so I do. I think my favorite medium changes over time. So all of the art that you've seen in this presentation was mine, unless otherwise labeled. So it depends on the day. Right now, I've been really leaning into digital art, which I do on my iPad, and that was what the Meadowlarks were done. This yellow-breasted chat is colored pencil, but the rest of the yellow-breasted chats you saw were digital. I think I like digital art because it's much more accessible to do. I can do it at any time. I can do digital art when I'm out in the field, studying birds. Taking 200 color pencils with you into the field is a little difficult. I have done that. But yeah, it does change a little bit depending on what bird I'm working on. But yeah, digital right now is my favorite, or watercolor. That's hard to say.
We have a few more minutes left. Another question in the room?
Thank you for a great presentation. That was wonderful, and a nice diversity of different things that you talked about.
Thank you.
My question actually came up during the question session. Everything, pretty much all the species you talked about are migratory species.
That is true.
So they are leaving these areas and coming back. And we've really focused on the breeding area, because that's when hybridization occurs. But does this mean, particularly with the chats in that tiny area, does that mean that they have like really solid phylopatry? They're coming back to exactly where they are born? What's happening with the other hybrids? I'm just wondering about that migratory and returning pattern and that aspect.
That's a great question. So there was a previous study that was done on yellow-breasted chats in a small population in British Columbia, where they have an isolated population up there. And they put little geolocators, little tiny backpacks on chats to look at where they migrated to. Because they were in the West, these yellow-breasted chats migrated all the way down kind of the coast, some through like Idaho and California, and then they settled into their wintering range down here on the western coast of Mexico. And it seems like that, along with some other data that they looked at, which is called stable isotope data, looking at what is in the feather when it was grown. Based on ratios of hydrogen isotopes, they found that eastern birds tend to just migrate to parts of eastern Mexico, along the Gulf of Mexico there. And western birds tend to just migrate to parts of western Mexico, and they don't seem to really interact with each other a whole lot. Like they have separate wintering grounds and separate migration routes. But the question of where hybrids go is so fascinating to me. And it's actually so fascinating that I tried to convince my partner to study yellow-breasted chat migration because he's a migration PhD student working on snipe. And his advisor did not bite on that research idea, so we won't know. But I don't know where they go. So there are other studies that show that hybrids of other species aren't as good at regulating their body. So sometimes those hybrids show up in places where they shouldn't. Sometimes they just die because they can't regulate their body temperature as well. And sometimes they have completely intermediate migration routes between their two parental species. So it could mean, because the wintering range of yellow-breasted chats is fairly small, that they still end up in areas where they can survive throughout the winter and where they have food they can eat. But we don't know. And I did actually try to get a collaborator that we have who's in Oaxaca and Veracruz right now catching horned larks for me. I did try to get him to catch some yellow-breasted chats down there, but he's too busy catching my study species now. So hopefully one day we'll be able to answer that question though. Of where did these birds go? It's an open question, so we'll see.
Sandra, any more Zoom questions? And then we'll have one more in the room.
I think our curiosity is sated for now and also spurred for future chat and meadowlark observations.
Okay, we have one more in the room.
Were you able to illustrate how the birds see each other, the colors?
I wish, but no. It's hard for us to fully imagine what they actually look like. I wish I could have. So there are people who have cameras that can capture the UV spectrum so that you can actually take a picture or a video of what things look like based on each other's vision. And there are people working on that. I wish I could have done that. But even with illustration, it would be pretty hard to be able to fully tell. And I would probably want like a full body scan of a bird to see really what the whole bird might look like under a different vision. So maybe one day.
All right, any last minute questions? Okay, I think our curiosity is sated in the room too. Thank you, Roomers and Zoomers, for all the great questions. And Johanna, what an amazing talk. Thank you.
Thank you guys so much.